there's so much knowledge on the shelves that no one applies because it is too complex to understand.
Welcome to the Greener Grass podcast from BlueBird Botanicals. I'm your host, Lex Pelger. If you're into deep science, you are in for a treat. I've sat down with a lot of the major scientists in the field, and I have to say that today's guest, Dr. Linda Klumpers, is one of the best I've ever heard for making the complicated biochemistry understandable. We'll hear about her research with synthetic cannabinoids, learn about the intricacies of working with pharmaceutical companies doing drug development, and find out about exciting new avenues of research. For all you armchair cannabinoid chemists, get ready for some fun. This show is brought to you by BlueBird Botanicals, to spread education about cannabis and other things on the greener side of life. BlueBird CBD oil comes from farms in southern Colorado and is grown using only water, soil, and sunlight. Go to bluebirdbotanicals.com for more info. Hello, everybody. I am here with Dr. Linda Klumpers, who has been kind enough to come into BlueBird Botanicals to talk about her work as a scientist and a cannabinoid studier.
Thank you for the introduction, and thank you also for showing me around. I really like seeing your facilities.
Oh, thanks. Yeah, it's a nice happy flock around here. And so you've done a lot in this world, all over the place. It's really fascinating. And so I was wondering, when was it that you knew you wanted to get into science, especially science at such a basic level?
Yeah, that's a very good question. To be very honest, as a little child, my mom always told me that I didn't have to ask so many things and look things up. In books, of course, because it was the early 1980s. And when I grew a bit older, let's say on elementary school, I knew that I wanted to be a researcher. I just didn't really know exactly the area, but I always was interested in biology a lot. And then by a process of choosing things that you really like to do, but also choosing for things you do not want to do, you eventually end up somewhere. And in my case, that was brain research. So I studied neurosciences.
And so how did you decide on that in the course of your university studies?
It was actually very difficult for me to come up with what study I had to study, because at the same time, during high school, I was very active in playing violin as well. And conservatory was tempting for me, not necessarily for violin, because I ended up conducting next to my PhD studies and work later on. But I was always fascinated by behavior of people, and not necessarily only the psychology. I really wanted to know how it worked. I was interested in consciousness. I was interested in embryology and biology. And eventually that is what led me to thinking, I want to be a brain researcher. So that is where that started. I must say also that during my neurosciences studies, there was a lot that I liked to do, but there were also a lot of things that I did not like. I really had a very, very hard time with all the animal research that was done. And it's not that I am against animal research or so, but for me personally, it was a very difficult thing to see. I was playing with the rats because I thought they were cute, and I didn't want them to be decapitated, as they call it. And that's when I decided at some point, I'm actually not sure if this is what I want to do. I'm not sure if I want to be a scientist. I was not really happy in the academic world. So after my master's, I actually made music for full time for 2 years before I went on with a PhD. And the PhD was not in mice anymore and not in rats, but it was in humans. So.
And before I ask about that, what was it like to be a working scientist who was also doing so much on the creative side? Because it's not the most common thing.
That's right. And it's also not really something that people always understand, because I saw that a lot of biologists like to hang out together. A lot of the musicians like to hang out together, and I hang out with both groups. But what they do is that is their whole world, and they do not only always understand that there is another world on the side. And for me, I just did my own thing. So during the week, I was mainly being the biologist, and on the weekends, I was making music. But at some point, actually, the two paths came together. Because after doing a very interesting, a big internship, I still had to write my final thesis. And the internship was very interesting, but that was the part I had difficulties with, because that was working with very, very young rats. It was on neurogenesis, very interesting. So neurogenesis is the development of new brain cells in the adult brain. And when I started, it was a very new discovery that adults actually have new brain cells that are being developed, because it was always thought, okay, if you have a cut in your skin will heal. If something happens in your brain, nothing will heal again. And that is true for a certain part, but it's not completely true that no new cells are being born. So it was a very, very interesting internship, but where the fields of music and biology came together was actually in my final thesis at university. For my master's, I decided I do not want to work with animals anymore, even though it was really interesting. The group that I was working with at was really good, but at some point I also just wanted to do something that I felt personally completely good about. And then I came up with the topic, I want to know more about what is happening in the brain when people listen to music. And that was already a topic that I tried to narrow down a lot, because you have the aspects of listening to music, you have the aspect of making music and composing music, and all these three things I thought would require different activities from your brain. So I wanted to study this, but the problem was that at that time, now it does exist, but at that time at the university I was studying, the University of Amsterdam, did not have any group that was specialized. So I found a professor in, from the Dutch Brain Institute that wanted to help me, and a professor from the Psychology Institute that wanted to help me. And this is how I could still write my final thesis about the biological processes involved in listening to music. And later on, a new institute actually was made from the musicology group combined with psychologists and biologists. And now there's a lot of brain research on music, actually. So yeah, that was a very good closure of my master's, actually.
It seems to really come together perfectly. You finally got the art and science to merge.
Yeah, that's right. That's right. I was very happy about it. It was, of course, tough because if something like that doesn't exist yet where you are working, then you need to create it yourself. And sometimes that's also good. It can be tough, but if you don't do it, then who will do it? I knew there was a group in Canada working on it, but I actually never got in touch with them. Maybe I should have, but I was very young, and everything is a learning process. One of the reasons why this whole field for me is so fascinating is that if you look at all the people on Earth, all the different cultures and all the different cultures, peoples that have been separated throughout the ages, they all have things in common. And the things they do have in common are language and music. Why does every people, every tribe, everyone have music in their culture? It's a very deeply embedded thing. If you see people suffering from all kinds of brain injuries and they're not able to talk anymore, many people are still able to sing. Many people forget words. They always remember the melodies. It's a language. It's a social bonding. It's just a beautiful thing, and I think it's really, really important to study this well.
That makes sense. It's one of the oldest ways that humans have to reach a spiritual state. And to get back to the neurogenesis, the funny part is, we didn't know that the brain was creating new cells until recently. And then it turns out that the cannabinoids seem to be creating new brain cells. So does the classic psychedelics. So does ketamine, it seems like. And these drugs that were so reviled turn out to somehow be helping. And all of those are used with music as well. Music helps enhance whatever you're doing. And to see that nexus is fascinating.
Yeah, at some point everything comes together and is related, and it's very, very interesting to study all the relationships and see patterns and structures.
And that seems to be how you went on with the PhD as well, because you got a PhD in clinical pharmacology, which I think is such a fascinating field because you're dealing at such basic level biochemistry science and then also looking at patients and real people and what's happening. So can you tell me a little bit more about your work then?
Sure. So first, because maybe not all of your listeners are very deeply, let's say, embedded in the science, pharmacology is the study of drug action, and clinical pharmacology is that means that you study drug action in humans, because you can study drug actions in cell cultures or in animals or tissues, and my specialization is actually to study how drugs work in humans. So that was actually a very logical step for me to take. I was very interested in the brains. I did not want to work with animals. I cannot cut in humans and see what's happening unless I work with dead brains. So I made the step to clinical pharmacology, to be very precise, clinical neuropsychopharmacology, meaning I was looking at the drug action in the brains and the nervous system in humans. So that is what I started doing. And when I started my studies in 2006, there was a very popular field, namely the field of the cannabinoid antagonists. And that is how I got involved in the cannabinoid system. So when in the 1990s the endocannabinoid system was discovered in organisms, at that time there was a thought, like let's say if a drug works in a body, very typically it works via a particular binding place. We call those binding places receptors. And a receptor receives a molecule on there, and that molecule or compound can have an action that stimulates or that does nothing or that inhibits. There are many different actions possible. So when the endocannabinoid system was discovered with two of those binding places, they were called cannabinoid receptor type 1 and cannabinoid receptor type 2, the researchers thought, okay, so if we can stimulate these binding places, there must be a way also to inhibit them. And why was that so interesting? Well, the effects of cannabis are, amongst others, that people get munchies. So you can see the pharmaceutical companies think, hey, wait a sec. So if we can induce a feeling of hunger in people, then we should also be able to inhibit that feeling of hunger. That should mean that we can maybe fight obesity, and that means big money, of course, and lots of opportunities. And besides, if cannabis can also make people dependent, because let's face it, some people are dependent of cannabis, then if we make an inhibiting molecule, we can maybe also fight addiction. And so this is a very simplified way of me explaining that the pharmaceutical industry thought that they would have a blockbuster here. And those potential blockbusters were actually the drugs that I started working on in 2006.
But this would have already been when the Rhymno ban had already passed, correct?
Yes.
Can you talk a little about what Rhymno ban is and how that would have affected your view on your work?
Exactly. So the company Sanofi, at that time it was still called Sanofi Aventis, they were amongst actually quite a lot of pharmaceutical companies that developed a drug in this particular class, in the class of cannabinoid antagonists. Sanofi Aventis had a few of these cannabinoid antagonists at the time, and the first one that they got on the market was a drug called rimonabant. This drug, to my honest opinion, I think was pushed to the market too fast, in way too much haste, and I think that to guarantee a better tolerability or safety profile of this drug, better research should have been done on the dosing and maybe on other compounds as well. And that's what you can see in at least some of the work that I did, where I compare rimonabant with other antagonists. Yeah, so rimonabant was brought to the market in Europe. And the FDA said, well, actually, we don't want to look at this yet because of the safety profile. So what was going on with rimonabant? Rimonabant, let me explain it this way, just in a bit of a common sense way. If you have cannabis and it induces the munchies, that's not the reason why people use it. The reason why people, tell me, Lex, why do you think people use cannabis?
That's the great part about it. There's so many reasons. Just because everyone has so many CB1 receptors over these different parts of their brains, I think it really helps to explain why some people get happy and some people get sad and some people get sleepy and some people get energized and some people get the munchies. And I think it's one of the most fascinating parts about it. And that's why it's so important to have researchers like you creating more nuanced, specialized molecules that can go in and do a more specific task than the typical cannabinoids, which do perhaps too many things across the body and brain for a doctor who wants to see an exact effect for a sick person.
Yeah. You said very good things there. You said precise effects, etc. The difficulty about the cannabinoid receptor, the difficulty about the endocannabinoid system is that it has two receptors only. I say only because there are many other physiological systems in the body, like the endocannabinoid system is a physiological system. You also have, for example, I'm just giving an example, the serotonin system, the serotonergic system. The serotonergic system has way more receptors. That means if you want to have specific effects, you have many more possibilities to target a particular receptor. With the cannabinoid system, that is much more difficult because you do not have this choice as you would have it with the serotonergic system. So, what happens if you use cannabis? A wide variety of effects happen. In general, people like it for the euphoric effects. For some people, it's a side effect. For other people, it's a desired effect. The euphoric effect, okay, so let's stand still with the euphoric effect for a second. Because if you make people happy by giving them cannabis, now we give this molecule that is, let's say, blocking the cannabinoid receptor or even giving inverse effects, that means that it can have consequences for how people feel. And that is actually what happened. Rimonabant did not only block the effect of the CB1 receptor, it also gave inverse agonism, meaning it gave opposing effects of what, for example, THC would do, which is a partial agonist, to get technical. So, and that is something that I think the researchers were either too optimistic about or they overlooked the problem. The problems with this. So what happened was rimonabant was brought to the market way too fast. People who were vulnerable to it would feel more depressed after using rimonabant chronically. They would even feel suicidal. And there were a few cases of suicides in the treated group with rimonabant versus the placebo group. And that is why the FDA actually never wanted to take it further into consideration and they never approved the drug. In Europe, what happened after that time was that it came to the market because, let's say, the European FDA, to call it that way, have a very different way of looking at drugs and food than the FDA has. At some point, they changed the prescription to can only be prescribed to people who never had depressive episodes before. And yeah, what happens in practice is that does not always work that way. If you tell people to do one thing, it doesn't mean that that is what they do. So after seeing the results from rimonabant being on the market in Europe, at some point the authorities decided that it had to be retracted, and rimonabant was withdrawn off the market.
It is unfortunate they rushed it so much, because that seems like it was a really important event in cannabinoid research, because it was a ship wrecked against a rock. And I think it scared some of the other pharmaceutical companies in industry who had cannabinoid research going, and then they wanted to shut down their shops because of that. And it's too bad, because the manipulation of the endocannabinoid system by pharmaceuticals, it's controversial to a lot of activists, but to have a more focused version of all the positive things that cannabis can do is going to be one of the most vital fields of medicine, probably coming up in the next couple decades.
Yeah, what you say, I agree. It's so extremely unfortunate what happened. It didn't only scare pharmaceutical companies, but it was also literally impossible to go on. So, for example, at Sanofi, what happened is that they had to start a dedicated department answering questions from the regulatory bodies. I needed information from them. I couldn't get it because they were slammed with work, slammed with questions they needed to answer. And I think it's very, very unfortunate. I think this drug was pushed too quickly, and I think the stigma is just too big, because now with this new revival of cannabis research, I do get a lot of requests from people asking me, don't you think it would be a smart idea to have cannabinoid antagonists? And I say, well, that's actually what has been going on over the past decades, but it didn't work out, because people now have a big bias against this. They think it's all evil, whereas the point I tried to make in my PhD thesis is, no, wait a sec. Of course, we need to be careful, but I think the wrong research was done and the wrong decisions were made in the whole process of getting this drug to the market. There are other options as well. We might come back to that later.
I was going to just go through your research a little bit chronologically, just because it seems to work so well that way. Because some of your first papers were on Namosol, which is a THC tablet, and you studied the pharmacokinetics, which is how it moved through the body. And so what was it like to be doing that research, and what did you learn? Yeah,
Namosol is a tablet that was developed by a small company called Echo Pharmaceuticals in the Netherlands. It was a THC tablet, and the novelty of that tablet was the enhanced absorption. So the idea is you take an oral tablet, but still it works really quickly in the body. It was a first in human. That means that it was never studied in humans before, and that means that you have to very carefully consider what dose do we start with and how many people are we starting it on. What a typical procedure is in drug development is that if you go to humans for the first time after you've done animal studies, is that you start in very small healthy volunteer groups. It's called phase 1, where you study they call it safety. I don't like to call it safety. I'd like to call it tolerability. If you want to see if something's safe, then you're doing something wrong. So we start looking at tolerability in that group, and we see, okay, how high can you go until you see adverse effects, so meaning effects that people don't want to have, like a headache. I'm just making something up. Also, even that I think is very unethical to do. You should not want to be looking at the tolerability. You want to know where is the therapeutic, let's say, limit. Where is the what is the dose that we have to give therapeutic effects? You actually don't want to be at the tolerability level, because then you're already too high. And for some drugs, there's a very small margin between therapeutic efficacy and safety. And for some other drugs, it's larger. But I think that if you set up your study in a really smart way, you should be able to already look at efficacy in these small, healthy volunteer groups. So that is what we try to do in that study.
And so, in addition to looking at how the drug was moving in, you were also doing fMRI studies at the same time and actually looking at how the brain was changing with THC.
That's right. The fMRI studies were done with an inhalation of THC. So now we move from the oral administration to the inhalation. There is... In early human drug development, there are different fields what you're looking at. I just mentioned tolerability is a very important field. The other two, you also mentioned one, is pharmacokinetics. How does the drug actually move through the body? How is it absorbed? How is it metabolized? So that means that it's being biotransformed to be excreted. How is it distributed? Where does it go? So, for example, cannabinoids are very lipophilic. They go to your fatty tissues, to your brain, etc. And then there's also the field of pharmacodynamics. So, what does the drug actually do to the body? What are the effects? So these are positive effects or can be adverse effects. And with the brain imaging study, what we wanted to do is to look at, okay, what is the relationship between how we measure these THC and metabolite molecules in the blood versus what do we see real time in the brain? What a traditional way of doing neuroimaging is that you put people in a scanner, you scan their brains while they do tasks, and then you see what changes in their brain in terms of brain activity. People call it brain activity, but in fact what is being measured mainly is a blood oxygen level. You look at where is the oxygen going in the brain, and you relate that to activity. We tried to do it in a different way. We put people in the scanner and they were not allowed to do anything other than look at the cross. That was just to keep them busy and not to have them fall asleep. They were just looking at a cross, doing nothing, and we wanted to know, are we able to measure brain patterns without them letting them do anything, but only by looking at the difference? We don't give them anything, let's say placebo, or we do give them THC. And we did find some differences indeed in the brain that we could statistically correlate to the THC levels in the blood, and to some effects also that we measured right after the people came out of the scanner. So, for example, how high do you feel? Or how was their postural stability and things like that.
And it did seem like one of the novel parts of what you were doing is looking at that resting state network of what a basic brain looks like, and then what happens when you change it with THC.
Exactly, because imagine if we would give them THC and let them do a task, then there can still be influence of that task to what you measure. And we didn't want to have that. We really wanted to look at resting state. Yeah. Mm-hmm.
And how were they inhaling inside an fMRI machine?
Yeah, that was a bit of logistics. We had to have them inhale outside of the machine, outside of the room even, because these imaging, the scanners are very magnetic. So you cannot have anything with a little bit of, magnetic force, let's say, inside the room. So we had to have them inhale outside the room and then immediately go into the scanner.
Those are some good volunteers dedicated to science, to take your high and then to go into one of those crazy machines moving stuff.
Yeah, we had great volunteers because, to be very honest, of course, THC can have very negative effects as well. People can get anxiety, and that is certainly something that happens. Imagine in this claustrophobic, for some people, environment, a scanning coil is very narrow, and that was not always pleasant for people. So we were very, very thankful for the volunteers.
And just to clarify, you talked about fMRIs and that they're measuring blood flow state. Can you talk a little bit about the controversy around the concept of fMRI and how much we can correlate neural activity to blood flow?
Yes, that is right. Although I must say that there are a few things improving in this field as well, if I can share that. So the techniques are getting better and more precise. And on the other hand, There are new techniques developing, and what is really fascinating to me, and there are some researchers working on this, and I encourage them so much, is that different imaging techniques are being combined. So in that way, you can combine, for example, anatomy with the direction in which specific signals are moving, and local activity, and a lot of work is being combined. Over the past few years, there's been this evolution in imaging techniques and combining and learning more about that. So things are changing in this field, but of course, if you have very complex signals that you try to analyze and you have to correct for so many variables, for movement, for example, then what are you eventually looking at? What signal are you looking at? What is it? And yeah, there's a lot of debate around that, and that will be for a while.
It is a fun time to read all of these technical innovations coming out on just figuring this stuff out better.
Yeah, for sure.
And that seems to be part of the work that you started doing afterwards. That's when you got into the CB1 antagonist work. And so you could tell me a little bit about why Surinband, and what you're looking at with that CB1 antagonist?
Sure. Surinband was one of the other antagonists that Sanofi developed at the time. This was already when Rimonabant, the sister of Surinband, was on the market. Surinband was not on the market yet, whereas Rimonabant was. And this was actually an opportunity to look at the efficacy of Surinband in healthy volunteer groups. What I just mentioned about Rimonabant is that I think that it went to the market too fast with studies that were done in a way that could have been done better. With Surinband, we had a new chance, actually. I say we. I was not part of the pharmaceutical company, of course, but from a part of an independent research institute. And what we just also discussed is that you look at tolerability profiles, but you want to look at efficacy profiles as well in healthy volunteers. That's difficult. If you are not sick and you're giving medication, then how are you measuring if someone has positive effects of that? Well, in the case of how would you do that with Surinband, you have a cannabinoid antagonist that you give to a healthy volunteer. Now what? So what we did is we did a THC challenge. That means that we gave the antagonist to the volunteers, but then we would challenge them by giving CB1 agonists and see, are these receptors being blocked by the antagonists or not? And is this all the receptors or not? And that is how we started playing with an antagonist in healthy volunteers. We gave them the antagonist, we gave them THC, and we looked at, do people still get high, for example.
That's cool. And so it did seem to show efficacy that was really working by looking at the people.
Yeah, it did show efficacy. And there are a few things that you can be happy about, and there are things that you can be worried about. Because if you know that this molecule is reaching the action site, you know, okay, we can have an effect here. This is maybe a very difficult story that I will not go into detail to. But to cut this long story short, it's also concerning that if you know that people do not get that high anymore if you give this antagonist, it's reaching the brain. And that means if it reaches the brain, that maybe it does induce depression or suicidal behavior in vulnerable people. And at the time, we expected peripheral action, so action outside the brain, to be beneficial as well. So, if this was a good thing, maybe, but it was definitely something we needed to study further.
And that's an intriguing part of the design, because your next paper was on TM38837. And it was a peripheral antagonist, which is just such a great concept. And I think it helps to explain to a lot of the activists why cannabinoid pharmacology is going to be so important. Is you're designing a molecule that wouldn't go into the brain, hopefully wouldn't have those depressive effects, but would still be doing some CB1 blocking on the various organs of the body where it might be helpful for certain things.
Exactly. And it was actually shown in studies with rimonabant that if you look at the weight loss in people versus the blood level changes, for example, for specific fat molecules, for cholesterol and so on, those improvements could not be attributed to the weight loss. So there was more going on, and it was very likely that that was happening outside of the brain. So yes, there was a lot to be potentially triggered there. After I did a serinabant study for the PhD, because this was all part of at least my PhD research, there was the big crash. The whole bubble collapsed of cannabinoid antagonism research. And my professors actually told me, well, Linda, maybe you should move to the GABAergic compounds because the whole cannabinoid antagonism market, let's say, collapses. And I thought, well, there must be different ways of doing this. And I started digging in molecule databases, and I found this company from Denmark called 7TM Pharma that had this molecule TM38837 that looked indeed like a peripheral antagonist. And I thought, well, maybe there's opportunities there. So I started talking to them at a conference, and indeed they were interested in doing a challenge study with this molecule. And right after I heard that rimonabant was taken off the market, I was informed by my professor. And we made sure to buy up all the rimonabant from the Dutch pharmacies. So we could actually do a direct comparison of this peripheral antagonist with rimonabant.
So you are professionally stubborn.
Well, I think sometimes you should just try a little bit harder.
And just for the nuts and bolts of a clinical pharmacologist, you have all these different molecules to select from. TM3837, a long string of molecules that people have been creating. How do you go about figuring out from the amount of data that's out there that this is one that you want to keep exploring? What are you looking for?
Yeah, it's not always easy to get all those data because typically it's all confidential data. You don't even know what the molecule looks like often. And it's sometimes not as easy to make your decision because you don't have all the information. You typically have to rely on what the pharmaceutical companies tell you. And at the time that this was all going on, the bigger pharmaceutical companies didn't actually want to take these risks anymore. So Sanofi was very clear, we don't want to continue in this field anymore. They had quite some molecules on the shelf. So serinabant we were talking about, the drinabant. Also other companies had a lot of molecules on the shelves. But I think that the smaller companies, are more risk averse.
And so you wrote these papers with many collaborators sometimes. You were usually a first author on almost all of this stuff, I believe. But can you talk about what it's like in collaborative science? I mean, you're responsible for the article. You're the first author, but there are all these other people working with you. What's it like to create the paper out of that data with those people?
That is a very fun process to do. Sometimes also difficult, of course, because there are many different opinions. But if you manage expectations from very early on and you're sure that you're all on the same line, it's only very helpful. Everyone has different expertise and everyone adds so much to that paper. A first author is really someone who writes it down typically, but it's really the work of a large group of people. And even though there might be quite some authors per paper, there were so many more people working on it than only the small selection of authors. So if we just step away from the writing itself, but look at what does a team look like in, for example, clinical pharmacological research. Of course, you have a project leader. You always have, and it can be a project leader. It doesn't need to be, have a physician who is the medical responsible person. You can have statisticians and methodologists. Those people are very important to determine up front how many subjects do we need. They randomize the people and the treatments. They look at what tests we are using and the variability, and that is all dependent on the sample size. Then you have research nurses that carry out a lot of the research, also the test assistants. You have the people in the lab doing all the analyses. Well, you see that I've already mentioned way more people than fit on the author page. Also, of course, there is the sponsor. They have a very important role because these are the people not only financially supporting it, but they have the molecules. They have the studies that were done before you start doing it. Because if you go inside a human for the first time, you really need to know what happened in animal research, what happened in all kinds of in vitro research. So that is research that was done outside of living organisms. So there is this whole big team, and I already now know that I've mentioned that there are quite some groups I've not mentioned, but of course there's limited time for this podcast as well.
True. And if people want to review your research, there's a great paper that you pulled together. We'll put a link to it in the episode notes. Novel Approaches in Clinical Development of Cannabinoid Drugs. And so we'll have that for everybody to read, as well as all of these other papers. And so I just wanted to finish. So how did you move then from academia into more of the consulting and seems more business-focused work that came afterwards?
Yeah, so after I finished my PhD research, I actually wanted to see other parts of the world. I joined a strategy consulting company to see what other industries are out there and what other types of work is there to do besides doing project leading for clinical trials. I learned a lot from doing that, and it led to me working for startup companies, mainly in business development and clinical research. I must admit, I do not really like big corporations with very precise procedures in place. That's not really my thing. So that is why I moved on to startups and work for them. I learned a lot there, but I have always known that at some point I wanted to work for myself. And that is what I'm doing in Colorado right now, actually. So I started a company called Tamori Pharmacology, doing business as Canify. And what we're doing there is trying to translate all that theoretical work and the practical work I've been doing before, and trying to make all the science that I have learned about over the past time, and also that we've created with this group, trying to bring that science to consumers and to product companies, because there's so much knowledge on the shelves that no one applies because it is too complex to understand, for example, or it's behind a paywall. And a lot of patients, for example, they can't reach that. And what I'm trying to do with Canify is to make all this knowledge available in a way that people understand it and that they can use it for the way they want to use it. So with Canify, we developed a test. It's an online test. People can go online, fill in questions about their symptoms and about their personal characteristics. So, for example, you can say, well, I suffer from nausea, I am male, this is my experience with cannabis, or maybe I don't have any experience. And then the test calculates based on human science what cannabinoids to use, in what dose, and what administration form for people with the same profile as has been shown in clinical studies. So we try to compare your profile with what we know clinical studies have shown. And then we inform you about that. We give the references to the study so you can always check back, is this true what they say? And to make it a bit actionable, we will compare your profile with products that are out there. So there, of course, it gets a bit less scientific because product companies do not always disclose everything about their products. There are a lot of product companies who do a very good job on this, but not all product companies do. So we try to say, well, this product is 80% relevant for someone with your profile, and this product is 30% relevant for someone with your profile. So you'll see what products might help for someone with your profile, and that way we try to engage people more with the science behind cannabis.
That's good. So you're working as well with the people, and it's an excellent test, by the way. We'll put a link to it in episode notes, but I took it myself and found it helpful. But you're also working with companies trying to figure out what might be helpful for them. And so are you feeling more optimistic after this last decade or two of kind of a slowdown in cannabinoid pharmaceutical research in terms of actual products coming to market? Does it feel like things are brightening again?
Yes and no. I never like it when research decreases. I always want there to be more, even though maybe research might not always lead to the desired results for a particular hypothesis. It will always lead to something better. So I was actually disappointed, very disappointed that the research in the cannabinoid field collapsed a bit. But I'm very happy that the attention is increasing right now. I just really, really hope that governments and regulatory bodies will make it easier to do more research, because I always think, and of course I'm biased as a scientist, but more research is definitely needed to better understand it, to help more people.
And so a last question then. Say that we could land you a giant grant for doing more of the work that you want to do. We could fund a laboratory doing just the kind of work that you would love. What would be the first research direction that you would love to take things?
I love this question because this is one of the many questions that I asked to someone you might actually know, Raphael Meshulam, who is being considered as the father of this whole field. It's the researcher that, together with another researcher, actually defined the right structure of THC. It was already found in the 1940s, but they didn't have it right. He did it again. Meshulam did it again in the 1960s together with Gaoni. And I was so curious what he would think if he could study anything, what he would study. And unfortunately, the answer was a bit disappointing. He said something like, I would like to study anything, everything. And, well, yeah, okay. That's not too exciting. Personally, of course, I've thought about this question because I asked it to other people as well. And, not to be lame and say I want to study anything, there are a few fields that I am particularly interested in. And one of them is definitely the field of psychiatry. A lot of people are afraid to study the cannabinoid system in terms of psychiatry because of the risk. But you have to start at some point. Other things that I'm very interested in are very, very basic things, actually, because it is almost shocking how little we actually know. So, for example, in 2010, I gave a presentation at the International Cannabinoid Research Society conference about the fMRI study that I did. But I also presented something else, which was a finding that we didn't expect. The fMRI study had 12 volunteers. 12 is a very standard number, actually, to use in exploratory imaging research. And when I presented this idea to the scientific advisory board of the research center, I said, so 12 people, so I want to include 6 females and 6 males. And then they asked me, why females? And I said, well, because they represent half of our population. Okay. It was accepted, but then the trouble started because I included females. But one after another, they kept dropping out from my study. They often couldn't handle the doses. And the females got far more nauseated. They got much more anxiety. And that was problematic. And I have no idea why this happened, but I had to present this at a conference because I wanted people to know, I wanted to share this, I wanted to hear experiences. And after I showed this to the audience, there were a group of a bit gray doctors that came to me, older men, and they told me, well, we have known this for years. I said, okay, that's interesting. So where did you publish? No one did. There is so little known. Is there a gender difference? And then what is that gender difference? How come? Also, there's much more because people also believe that 11-hydroxy-THC, which is the primary metabolite of THC, is more potent. Oh, really? Is that true? It also has a larger brain penetration. Okay, I've seen that in studies, but what receptors does 11-hydroxy-THC work on? Try to find it. It's very difficult. Trying to find more about the safety profile of inhaling terpenes, metabolism of terpenes. What does THCA do in the body? Has anyone ever studied that in healthy volunteers or in patients? No. There's so much basic research that still needs to be done. So there's a lot.
And one I remember when I interviewed Meshulam, that's what he got all worked up about was anandamide, the neurotransmitter cannabinoid that he helped to discover, that it had never been given to healthy human patients in simply an IV form. When you have something like insulin that within 5 years was on the market, and all these other famous drugs, as soon as they were found to be components of the human body, they were soon being put into human bodies to fix them, and anandamide just didn't get looked at.
Wow. Yeah, there's a lot that hasn't get looked at, and I'm not a chemist, so I can't tell for anandamide, but I can see some challenges doing that because it has a relatively short half-life, so it means it's being broken down pretty fast. These endogenous cannabinoids, endocannabinoids, they are being made between neuron cells, between brain cells or nerve cells, and the whole idea of the cannabinoid system as it is today in humans is that it is there for homeostasis, so for keeping a balance in the body. They work in a retrograde way. That means that if let's say one cell is stimulated by another cell, it sends cannabinoids back to do something about the balance of the communication. And that means that they do not need to be long-acting. They don't need to go over large distances in the body. So I have no idea if there are practical challenges in getting this done or whether it's really lack of interest.
So many unanswered questions.
So many.
Dr. Klumpers, thank you so much and for coming in to talk to us, and best of luck helping to solve some of these problems.
Thank you. It was a great pleasure.
Greener Grass is a BlueBird Botanicals podcast. Their CBD oil supports a healthy body and a strong endocannabinoid system. They've got friendly customer service who can answer any of your questions, and the number is right there at the top of their webpage. But, per the FDA, they won't be able to make any medical claims for these nutritional supplements. That's also the reason you'll hear little about CBD on this show. There's no need for us to add more evidence about CBD when a simple Google search will give you more than you can read in a month of Sundays. So this show covers the cannabis world and more with editorial freedom from BlueBird Botanicals. Thanks for joining the Greener Grass podcast. As always, our audio alchemist is Matt Payne. The Gypsy Jazz theme music comes from Brett Van Donsel. Our beautiful bird sounds are courtesy of Lang Elliott. And I'm your host, Lex Pelger, wishing you a bright green day.